So for chapter 8, we'll talk about trophic levels and energy flow, the main point of photosynthesis, the anatomy of the chloroplast, the anatomy of a leaf, the stages of photosynthesis, and then we'll talk about the two big reactions, the light reactions and the Calvin cycle. And then we'll talk about some variation in photosynthesis with C3, C4, and CAN plants. So trophic levels are feeding or nutrition levels, and you probably remember talking about food chains before, and maybe talking about producers and consumers. An autotroph means self-nutrition. So autotrophs make organic molecules from inorganic sources. And most of the time when we're talking about autotrophs, we're going to be talking about photoautotrophs. That means they use light as a source of energy to create organic molecules from inorganic molecules. So photosynthesis is what we're talking about there. So green plants are probably what you think of when you think of photosynthesis, but also algae and some other protists, and then cyanobacteria can do photosynthesis. And then we've got heterotrophs, which obtain organic molecules from other organisms. So down on the bottom there, we've got a picture of an autotroph, which is that plant who made sugar, and you've got a moose, who's the heterotroph. So just like we talked about how it's important to understand like the big picture or the main point of cellular respiration, you need to understand the big picture or the main point of photosynthesis before you even worry about the nitty-gritty details. The whole point or the big picture on photosynthesis is that photosynthesis converts solar energy into chemical energy. So light energy is captured and used to synthesize carbohydrates. This is an inorganic redox reaction. So we've got carbon dioxide and water, and then the energy that's required comes in the form of light energy, and with the help of some enzymes and some specific structures in the cells, you get glucose and oxygen. So carbon dioxide is reduced and water is oxidized in this reaction. So I feel like when people think of photosynthetic organisms, everyone probably thinks about plants. Plants are not the only photosynthetic critters. In this picture, we've got a sunflower, that's a plant. You've got a deciduous tree, like a tree that loses its leaves, and an evergreen tree. Those are plants. Probably think of that. Moss, that's a plant too. Maybe you didn't know it, but it is. And then you've got a euglena, which is a protist, which is actually able to do photosynthesis, and depending on the species, they can be heterotrophic as well. You've got kelp, which is actually a protist, even though maybe you thought it was a plant before. And then we've got, a single-celled protist called a diatom, and then we've got a photosynthetic bacteria there as well. In order for photosynthesis to occur, the cells that are doing this series of reactions need to have a particular kind of structure in order for this to happen efficiently. So they have to have the right enzymes, they have to have pigments to capture light, and they have to have a membrane as well. So probably when you think of photosynthesis, you think of chloroplasts. Chloroplasts are structurally similar to photosynthetic bacteria called cyanobacteria, and they likely evolved from those bacteria. So we're going to focus on photosynthesis in a chloroplast, which would be how eukaryotic organisms like plants and some algae are going to carry out photosynthesis. So the chloroplast has an outer membrane, and then it has an inner membrane, and then it has a third membrane called the thylakoid membrane. That third membrane contains pigment molecules that absorb solar energy, and then when we have stacks of thylakoids, we call those granum. So then we've got the stroma, which is the liquid portion inside of the chloroplast. So this is kind of silly, but if you think of the granum as like a stack of pancakes, each pancake would be a thylakoid, and then the stack would be the granum, and if we have lots of stacks, that would be grana, which would be plural, and then the stroma, since it's the liquid portion, it would be like the syrup on the pancakes there. Just like with the mitochondria in cellular respiration, it's helpful to know the chloroplast anatomy so we can understand the location of the different reactions and understand the structure of the chloroplast and how that relates to the reactions that are occurring. So we also need to understand what the leaves are like in the plant in order to understand photosynthesis in plants. So the middle layer of the leaf is called the mesophyll, and that's where most photosynthesis occurs. And then the leaf also has pores for gas exchange. These pores are called stomata, and they allow carbon dioxide to go into the leaf, and they allow oxygen to exit the leaf. They also allow water to exit, which will be important if you go to Bio 170 and learn about transportation of water in the plant. So ultimately the water and carbon dioxide are going to diffuse into the chloroplast, and that will help facilitate photosynthesis. So you can see we've got a couple pictures. The pores in the leaf have guard cells that open and close the pores in the leaf, and we'll talk about that later as well. So how do autotrophs get carbohydrates? And what's an example of an autotroph? So remember, auto is self, troph is nutrition, so autotrophs can produce their own carbohydrates. So any photosynthetic critter you could think of would be an example of an autotroph. So like a plant, algae, alga really, or some bacteria as well. Or if you said like a rose bush or grass, none of that would matter. That would all work, and that would all count. Then how do heterotrophs get their carbohydrates? And give me an example of a heterotroph. So remember, heterotrophs have to have preformed organic molecules, so heterotrophs could either ingest them or absorb them depending on the types. Examples of heterotrophs include dogs, or we've got humans, or a cow. Those are all heterotrophs. Also fungi and some protists as well. Then draw and label a chloroplast. Include the stroma, the grana, and the thylakoid. So somebody from the internet drew one for me. Remember, the chloroplast has an outer membrane, an inner membrane, and the third membrane. So I described it before as pancakes, right? So the third membrane is like the stacks of pancakes. So each pancake is a thylakoid, the entire stack is a granum, and then all the stacks of pancakes are grana, because it's plural. And the stroma is the liquid portion around your membrane stacks. So when it comes to light, we can really think of light as the form of energy. If you take a physics class, they'll get into more details on that, but we can kind of think of a photon as a discrete bundle of energy. As far as wavelength and energy content go, violet light has a shorter wavelength and higher energy, and red light has a longer wavelength and lower energy. You've probably heard of ultraviolet radiation before, usually with regards to like damaging your DNA and causing cancer. It's usually what I think of when I think of UV radiation. And then you've probably heard of infrared radiation before as well. So not all light is visible, and there's a picture of the visible light spectrum and then other wavelengths of light as well on the figure on this slide. So after an electron absorbs energy, it's in an excited state and is usually unstable, and that can be released as either heat or light, and excited electrons and pigments can be transferred to other molecules or captured. So we'll talk about electrons being bumped up to a higher energy level or becoming photoexcited when a photon strikes that particular electron. So there's different pigments involved in photosynthesis. These pigments can absorb some light energy and reflect others. Leaves on most plants are green because they absorb red and violet light and reflect most green wavelengths. And having different pigments allows plants to absorb light at many different wavelengths. So 3 important pigments are chlorophyll A, chlorophyll B, and beta carotene. And you can see the action spectrum for photosynthesis down there, photosynthetic pigments, and you can also see the absorption spectra for the particular pigment in that first figure. So with photosynthesis, I think it's important again to think of the big picture on photosynthesis. Right, the whole point of photosynthesis is to produce a carbohydrate from carbon dioxide and water. And then from there, go on and break it into the 2 reactions, and then work on the reactants product's location and light requirement for these particular reactions. So just like with the picture of the mitochondria and how I told you before that would be good for understanding cellular respiration, this picture of the chloroplast is kind of the summary of photosynthesis. So we'll get into the nitty-gritty coming up here. So the light reactions happen when solar energy is available, right? They happen in the light. What a great name. They happen in the thylakoid membrane of the chloroplast. So that's the most interior membrane in the chloroplast. And they also require pigment molecules to absorb solar energy and boost electrons to higher energy levels. Going into the light reaction, we've got water, ADP, and NADP. We also have something called photosystem 2, which uses an electron transport chain, and photosystem 1, which reduces NADP. So we get oxygen, ATP, and NADPH, which is kind of like an energy carrier, which sets us up for the next reaction with the light reactions. There's a few videos I posted of it, and I really think it's worth watching these videos that show you the different molecules moving so you can get kind of an idea of how the light reactions actually work. But we're going to go from here and kind of walk through the rest of the slides. All right, so this shows you the organization of the thylakoid, so that inner membrane inside of the chloroplast. These yellow areas are supposed to be light coming in. Also notice there's this hydrogen ion concentration gradient where there's more hydrogens on the inside of the thylakoid space than the outside. So if we start with light striking photosystem 2, that is going to bump an electron up to a higher energy level, and that electron will get passed along an electron transport chain here, and as it's going along that electron transport chain, that's going to provide the energy to pump hydrogen ions across their membrane here against their concentration gradient. Once we get an electron passed on from photosystem 2, we get more electrons by splitting water here, and that's actually where we produce the oxygen. So after photosystem 2, the electrons get passed to photosystem 1, which is really an unfortunate naming system, right? Like 2 comes before 1 in photosynthesis. So photosystem 1 here has an electron now. That electron gets bumped up to a higher energy level by this light energy. Electron gets passed to NADP reductase, and that reduces NADP and produces NADPH over here. And then if we get back to that proton or hydrogen ion concentration gradient, you will notice that we've got an ATP synthase as well. And similar to the mitochondria where we had ATP synthase and a proton gradient, as hydrogen ions flow across the ATP synthase here, that's going to provide the energy to produce ATP from ADP and that phosphate group, just like water going across a water wheel or wind going across a windmill. So that's the organization of the thylakoid and the light reactions. So again, our chemical products of the light reactions include oxygen, which is produced in the thylakoid lumen, so the inner part of the thylakoid, by the oxidation of water by photosystem 2. And then NADPH is going to be produced in the stroma from high energy electrons that start in photosystem 2 and are boosted in photosystem 1. So we've got NADP plus electrons plus hydrogen gives us NADPH. And then ATP is produced in the stroma by ATP synthase using that proton electrochemical gradient. So where do the light-dependent reactions occur? The light-dependent reactions happen in the thylakoid membrane, and remember the thylakoid membrane is part of the chloroplast. What is one pigment that absorbs light energy? So you were probably thinking of chlorophyll, so chlorophyll A or chlorophyll B or carotenoids would work. And then what does ATP synthase do? ATP synthase synthesizes ATP. Remember that protons flow through ATP synthase and it turns the rotor complex just like water over a water wheel or wind over a windmill, and that provides the energy to produce ATP. So that brings us to the Calvin cycle. This is where we actually produce the carbohydrate in photosynthesis. I say we, obviously we don't do that because we're not plants or photosynthetic organisms, but I don't know, I guess we're like in the magic school bus or something. So anyhow, Calvin cycle is where the photosynthetic organism produces the carbohydrate. So these used to be called the dark reactions, but that was kind of a bad name because they don't really require dark to occur. Sometimes they're referred to as the light independent reactions because they don't require light to occur, but often we call them the Calvin cycle. This happens in the stroma, which is the liquid portion of the chloroplast. And going into the Calvin cycle, we've got carbon dioxide, and we also have the ATP and the NADPH that were produced in the light reactions. A product of the Calvin cycle includes glyceraldehyde 3-phosphate, and then we also get ADP and NADP, which can be recycled back to the light reactions. So I've got some videos that I posted on your Canvas page that I think will help kind of refine this and help you understand the Calvin cycle a little bit better, so take a look at those as well. So to make one G3P, the cycle has to take place 3 times and fix 3 molecules of carbon dioxide. The Calvin cycle has 3 phases: carbon fixation, reduction, and the regeneration of our carbon dioxide acceptor. So in the first phase, carbon dioxide is incorporated into RUBP, that carbon dioxide acceptor, using the enzyme Rubisco. And the reaction product is a 6-carbon intermediate that splits into 2 3-carbon molecules, which are 3PG. From there, we have the reduction in carbohydrate production. So that involves reduction and phosphorylation of 3-phosphoglycerate to G3P. And It takes 6 ATP and 6 NADPH in order to produce 6 molecules of G3P, but only 1 exit the cycle for use by the cell. Then we regenerate the RUBP, so you rearrange the 5 remaining molecules of G3P to regenerate that initial carbon dioxide acceptor, which is called RUBP, and that takes ATP as well. So if we look at the kind of figure of the Calvin cycle, you've got carbon dioxide entering in the Calvin cycle here. Rubisco is the enzyme that helps bind our carbon dioxide to our acceptor here, RUBP, and we get 3PG. From there, we spin some ATP and have an intermediate, right? And eventually we get to G3P. We keep 1 G3P, or the plant keeps 1 G3P. The other 5 G3Ps go back to build RUBPs, which are the initial carbon dioxide acceptor with that help of the enzyme Rubisco. So carbon dioxide is incorporated into carbohydrates, which are the precursors to other organic molecules in the plant, and also they're important for energy storage as well. This requires a lot of energy, like we said. It takes lots of ATP and lots of NADPH, and the end product of the Calvin cycle is G3P, which can be converted to other molecules like glucose. So that G3P can be converted into glucose phosphate, or sucrose, or starch, or cellulose, or fatty acids and glycerol, or amino acids. So this is kind of a cheesy diagram here, but you've got a tree and you've got your G3P, and you're getting to see how that G3P can be used to make all the other organic molecules in the tree. So where does the Calvin cycle happen? So the Calvin cycle happens in the stroma, and remember again, the stroma is part of the chloroplast. And then what's the main point of the light reactions? Really, the light reactions set us up for the Calvin cycle, or set the plant up for the Calvin cycle. So the light reactions produce the ATP and the NADPH needed to run the Calvin cycle. And then what's the main point of the Calvin cycle? So the Calvin cycle produces the carbohydrate, so G3P, or you could say glucose as well. So Rubisco is that enzyme that we just talked about, binding carbon dioxide with RUBP and producing 2,3-PG. Or Rubisco can also bind to oxygen, which eventually can cause a release of carbon dioxide, which is called photorespiration. This is more likely in hot and dry environments, and it's favored when carbon dioxide concentration is low and oxygen concentration is high. It's more likely to make the mistake if there's lots of oxygen and not much CO2. So, do you think photorespiration is good or bad for the plant? So, it's bad for the plant because instead of producing carbohydrate, it's actually losing fixed carbon. So environmental conditions can really influence both the efficiency and the way the Calvin cycle works. Environmental conditions include things like light intensity, temperature, and water availability. So different photosynthetic pathways have evolved to kind of take advantage of different parts of the environment or different environmental conditions. So we've got C3 plants, C4 plants, and CAM plants. C3 plants are named because the first detectable molecule after carbon dioxide fixation is a 3-carbon molecule, right? C3, 3 carbons. Most of the time when you think of plants, you're thinking of a C3 plant. C4 plants are named because the first detectable molecule following carbon dioxide fixation is a 4-carbon molecule. So corn, crabgrass, those are examples of C4 plants. CAM plants are named for the family they were first discovered in, so Crassulaceae is the family, so this is called crassulacean acid metabolism, or CAM. But it's not just found in that family. It's found in many succulents and then other plant families as well. In C3 plants, photosynthesis takes place throughout the leaf. Their stomata, those pores in the leaf, are open during the day, and under water stress the stomata close. And when the stomata close, that limits water loss, but also it limits carbon dioxide gain. So this reduces the efficiency of photosynthesis because oxygen is going to compete with carbon dioxide for the active site of that enzyme Rubisco. And so then fewer 3-carbon molecules are going to be produced because of photorespiration. So these plants are good in cool and moist conditions because they don't have to have any specialized anatomy and any specialized enzymes. So in conditions when it's not too hot and water isn't really limited, they're going to usually outcompete C4 and CAM plants. In C4 plants, photosynthesis takes place in the inner cells. C4 plants represent partitioning the reactions of photosynthesis in place. They do carbon dioxide fixation in something called the spongy mesophyll, and they do the Calvin cycle reactions in the bundle sheath cells. Their stomata are open during the day, and they avoid the uptake of oxygen by rubisco by increasing the amount of carbon dioxide that is available to the enzyme. These are best under high light intensity and high temperatures because the carbon dioxide is delivered directly to rubisco and not allowing rubisco to kind of accidentally grab oxygen. So this avoids photorespiration or limits photorespiration. They don't have to keep their stomata open as much then, so they're going to lose less water for the same amount of carbon dioxide gained for photosynthesis. So I know I like to give you guys pictures on the exam, but I'm not going to give you a picture of a C3 plant and a C4 plant and ask you to tell me the difference, but I just want you to look at the difference in their anatomy. Look at the arrangement of the mesophyll cells and the bundle sheath cells here. So in the C3 plant, the mesophyll cells are arranged in parallel rows, and in a C4 plant, the mesophyll cells are layered around the leaf veins, and then the bundle sheath cells are shielded from the buildup of oxygen, so the Calvin cycle is going to happen there. So that brings us to CAM plants, or crassulation acid metabolism. These plants do partitioning based on time instead of space. So all the other plants have their stomata open during the day, which makes sense because that's when light's available, so that's when it would make sense to do the Calvin cycle as well. But CAM plants open their stomata at night, which for them makes sense because you're less likely to lose water at night because night is going to be cooler, right? So you're going to lose less water there. So the carbon is fixed into a 4-carbon molecule, which is an acid, which is where the name comes from. And then during the day, the stomata are closed, which prevents water loss, and that 4-carbon molecule is going to release carbon dioxide to the Calvin cycle. So these plants are very good at conserving water, and some of them can even CAM idle in really dry times. They leave their stomata closed during the night and the day, and they can use the oxygen given off in photosynthesis for cellular respiration and the carbon dioxide given off in cellular respiration for photosynthesis. They can't do this forever. They're not like a perpetual motion machine or anything like that, but it can help them survive dry periods that other plants could not and give them an adaptive advantage in those situations. So this figure shows you a C4 plant, and it shows you the mesophyll cell and the bundle sheath cell of the C4 plant and how the reactions of photosynthesis are partitioned in space. And then we've got a CAM plant where they have a day version and a night version in the mesophyll cell, and you can see the initial carbon capture is at night, and then the Calvin cycle runs during the day when ATP and NADPH are available because they were produced by the light reactions. So go ahead and match these pictures to the particular type of photosynthesis. Pause it if you want and think of your answer and then hit play when you think of it. So most students think that 10 is probably the easiest one to figure out. So that happens to be C4 photosynthesis. It's showing you carbon capture is occurring at night. It shows you that the first detectable molecule is a 4-carbon molecule. And then probably we'll just go to number 11. Number 11 is C3 plants. There's a 3-carbon molecule right there that they're showing you. So that might give you a hint. Also, there's no real specialized anatomy in this particular case. And then with number 12, this is a C4 plant. You can see right there, it says C4. That's showing you the first detectable carbon molecule after a carbon fixation is a 4-carbon molecule. We also have the mesophyll cell and the bundle sheath cell where the Calvin cycle occurs in the bundle sheath cell. So that's the different kinds of photosynthesis. One thing to make note of that drives me a little bit bonkers is that people will tell me that CAM plants, like in number 10, do photosynthesis at night. Think about that for a second though. Can a plant do photosynthesis at night? What is photo, right? Photo is light. Synthesis. So using light to synthesize an organic molecule cannot happen at night because there's no light at night. So be careful with that. They don't do photosynthesis at night. What they do is they capture carbon at night, or they fix carbon at night. So here's another misconception that students will tell me. They will say in photosynthesis, plants convert carbon dioxide to oxygen. So think about that, pause it while you're thinking, and then hit play when you're ready. Okay, so There's a lot going on with that, right? So where's the oxygen come from? And what happens to the carbon dioxide? So the oxygen comes from the splitting of water in the light reactions, and the carbon dioxide is part of the Calvin cycle. So that's a problem right there, right? So the oxygen does not come from the carbon dioxide. The oxygen comes from water, and it's produced in the light reactions. The carbon dioxide is used to create carbohydrate in the Calvin cycle. So be careful with that thought as well. So that's the end of this one. We talked about trophic levels and energy flow. Remember, we've got autotrophs and heterotrophs, and in particular photoautotrophs. Remember, the whole point of photosynthesis is to capture light energy and produce organic molecules, so to make a carbohydrate. We talked about the chloroplast anatomy, so the thylakoid membrane, the stroma, the grana. We talked about the leaf anatomy, so the mesophyll and the stomata. Don't confuse stoma and stroma, or stomata and stroma. One is part of the chloroplast and one is part of the leaf. We also talked about the light reactions and the Calvin cycle. So focus on the reactants and the products. In the light reactions, we talked about photosystem 2 and photosystem 1, and ATP synthase and NADP reductase. So be aware of all of those as well. The light reactions include, ATP synthase and a proton gradient, which is very similar to the mitochondria and how they produce ATP. Then we took the ATP and NADPH produced in the light reactions and went to the Calvin cycle, where we produced a G3P, and 2G3P can make a glucose. And remember, you have Rubisco, which is the enzyme that binds the carbon dioxide to RUBP and gets the Calvin cycle started. And then we had variation on photosynthesis in C3, C4, and CAN plants. And sometimes that Rubisco can make a mistake and bind oxygen to RUBP instead of carbon dioxide, and that leads to photorespiration, which is a bad thing, and that's what the C4 and CAN plants are trying to avoid. So that's the end of this chapter.